when we looked at the brain gene activity profiles of these so-called precocious foragers, we found that the profile is very similar to a normal age forager. In other words, the environment that induced the change in behavior was acting to change the activity of the genes. So we found evidence, some of the very first evidence for extensive environmental regulation of brain gene expression. And so that we just saw the changes in gene expression, and that meant we needed to start looking for what are the actual molecular changes that are occurring. And then you'll recall I said it's nurses growing up to be foragers, can't be a change in DNA, it's the same bees. So going back to the very first comments I made, it means these are epigenetics. So we got into epigenetics by the phenomenon of environmental changes in gene activity that cannot be explained by changes in DNA sequence, therefore must be changes at the epigenetic level.
Hello and welcome to the Moral Medicine channel. My name is Eric, and today I have the honor and privilege of interviewing a very special guest. So Dr. Gene Robinson is an American entomologist, director of the Carl R. Woese Institute for Genomic Biology, and National Academy of Sciences member. He also holds affiliate appointments in the Department of Cell and Developmental Biology, the program in Ecology, Evolution, and Conservation Biology, as well as the Beckman Institute of Science and Technology. He received his PhD from Cornell University, and he has pioneered the application of genomics to the study of social behavior and led the effort to sequence the honeybee genome. Recently, Dr. Robinson has devised a new approach to 3D imaging that captures DNA methylation, which is a key epigenetic change associated with learning in the brain. Thank you so much for joining us today, Dr. Robinson.
Thank you for the kind introduction, and it's a pleasure and an honor to be here with you and to speak with through to the entire group.
Thank you so much. So, Dr. Robinson, I thought it would be a great starting point to begin by asking you, just for the layman in the audience, what are epigenetics and how did you get involved in this branch of science?
Yeah, that's a really good place to start. So epigenetics is a term that refers to, if you dissect the actual word, epi means on top of, and then genetics refers to genes or the genome. So it connotes the concept that there are molecules that can be added to the DNA as the fundamental substance of genetics, which forms our genome. These are substances, chemicals that can be added to the DNA that modify the actions of the genome without changing the sequence of the genome per se. So the more traditional way of thinking about how the genome works is if there are changes to the DNA, mutations is the technical term to the DNA, that results then in a different configuration of genes, a different level of activity for a gene, a different version of a gene, inactivation of a gene. All of those things can happen when you change the sequence. But lots of other stuff can happen if you leave the sequence alone and then just add molecules to attach to the DNA, which then modify the actions. So the science of studying those chemicals, who they are, where they attach to the DNA, and then what they do, all of that is encompassed in the term epigenetics. And the way my lab got involved in this is my lab studies honeybees. And just over 20 years ago, we started some of the very first experiments in the field to look at measuring all of the genes in the brain, the activity of all of the genes in the brain as a function of behavior, their behavioral state, and also the environment. So it was just brand new technology made possible by the burgeoning field of genomics, and the sequencing of the honeybee, as you mentioned, made it possible to ask these kinds of questions. Prior to that, one could measure the activity of genes, but one at a time. And so we did, as a field, already know that there's some plasticity, some flexibility in the activity of genes, but we had no idea the extent of it because it wasn't possible yet to do the large-scale experiments. In the early 2000s, they became possible, and we performed some of the first experiments using honeybees. And what we found, so we compared two behavioral groups. We compared bees that stay home, work inside the beehive, and take care of the baby bees, and those are referred to as nurse bees. And then other bees that go out, the ones that you're familiar with, you see on flowers, those are the foragers. So same beehive, same mom, the queen, same environment there. And we compared the activity of the genes of nurses and foragers. And we were absolutely struck by the finding. The finding showed that 40% of the genes showed differences in activity coming from the brain of a forager or a nurse, 40%. So the changes are just really extensive. And then I did not tell you something on purpose in order to make the point now. Nurses are younger bees, and they grow up and become foragers. So it's the same bees, same genetics, nothing changing in terms of their DNA sequence, but what they're doing is associated with a dramatic change in the activity of the genes in the brain. So that was the first result. Second result was we took advantage of the fact that honeybees have a very flexible division of labor. So some bees are nurses, some bees are foragers. They're nurses when they're young, then they grow up and become foragers. But if you stress the colony, they can respond to changes in the labor market. So if you remove the foragers to simulate, say, a storm or a predator that decimates the foraging force, the colony just doesn't sit there and perish. They actually spring into action in response to that perturbation. And what do? Some of the young bees grow up faster and become foragers at an age much younger than they would under normal conditions. So adult worker honeybees live about 6 weeks. They spend the first 2.5 to 3 weeks inside the hive, and then they graduate and become foragers. So when you see a bee on a flower, she's at least 2 to 3 weeks old. They live about a week as foragers, and then they die. So if you remove the foragers, you can get some bees starting to forage as young as 1 week of age. So an environmental perturbation. When we looked at the brain gene activity profiles of these so-called precocious foragers, we found that the profile is very similar to a normal age forager. In other words, the environment that induced the change in behavior was acting to change the activity of the genes. So we found evidence, some of the very first evidence for extensive environmental regulation of brain gene expression. And so that we just saw the changes in gene expression, and that meant we needed to start looking for what are the actual molecular changes that are occurring. And then you'll recall I said it's nurses growing up to be foragers. Can't be a change in DNA. It's the same bees. So going back to the very first comments I made, it means these are epigenetics. So we got into epigenetics by the phenomenon of environmental changes in gene activity that cannot be explained by changes in DNA sequence, therefore must be changes at the epigenetic level.
Absolutely fascinating. And the next question, you already answered it in a sense, but I do want to dive further into this topic. So how do external factors influence the expression of genes?
Yeah, so I'll frame it in the context of the brain and behavior, which are my specialties. So another way to ask the question is, how does the environment get under the skin? We know that we as humans and all species are exquisitely sensitive to changes in the environment. In the context of behavior, it's very important to have a system that's responsive to the environmental conditions. So in the context of behavior, stimuli are perceived by our sensory systems, and then they are processed to be able to provide information, directions, if you will, instructions that can lead to adaptive changes in behavior. So take the example that I gave before. We delved into this and found, so we can ask the question of like, how do young bees know that there are no foragers? What's going on here? And the question in honeybees is very interesting because the honeybee colony has no centralized control. It is decentralized. One might think, oh, what about the queen? There is a queen for sure. Everyone knows that, but she doesn't tell the workers what to do. So it's not like she has superior abilities to sense the demography of a colony and say, oh, geez, we're short on foragers. Why don't you, and you start foraging at a younger age? Doesn't work that way. Human labor markets are somewhat more centrally organized. In the US, for example, a well-known business newspaper, the Wall Street Journal, might cover labor changes. And there might be an article that says there is a shortage of medical technicians. How do they get this information? They acquire this from a variety of sources. They synthesize it, and then they say there's a shortage. And that will prompt universities, technical colleges, individuals to say, maybe I need to change my behavior in some way to respond to that. Honeybee colonies, there's no such centralized control. The individuals are somehow picking up local information to get that initial information about the environment. And we have studied this for some time and found that there are chemicals produced by foragers that inhibit the maturation of their younger sisters. So the more foragers that are there, the more chemical inhibition there is. So the young ones stay young and take care of the baby bees like they're supposed to. If you remove those foragers, you have also removed the inhibitory chemicals. And in the absence of that inhibition, the young ones grow up faster. So that's the first stage. So there is a environmental signal, and environmental signals could be visual, could be auditory, could be mechanical, or could be smell. In this case, it's a smell. It's a chemical that the bees perceive or perceive the lack of, and that's the first step. We have found that those kinds of chemicals act on the genome in the brain to shift the gene expression, gene activity profiles more towards the forager. So they inhibit, and so they'll keep the nurse state. So they encourage the nursing brain gene expression state. You remove the pheromone, the chemicals, they're called pheromones.
Mhm.
You remove those chemicals. In the lack of that inhibition, the gene activity profile becomes forager-like. So that's the path. And those are the kinds of pathways that occur in many other contexts. You start with a sensory and external signal, some sort of perceptual ability to get that information, to get that inside the organism. The brain processes it, interprets it, and then directs activity. So you have neural control and molecular control. Then the molecular control feeds back to change the neural control. So genes don't forage, they orchestrate changes in the brain, in the nervous system, neural activity to change muscle activity so that the bee will forage.
Absolutely fascinating. Of course, I'm not a scientist, but I'm really curious to get your thoughts on something that this reminds me of to a certain extent, which is the returning soldier effect. Maybe there's absolutely no relevance at all here, but when you talk about how certain external factors can influence the bee population, it does remind me a little bit of the returning soldier effect, in which scientists have observed that the number of boys who are born after and during wars is much greater than during times of peace prior to war. And this is something that has been replicated. I'm just curious to get your thoughts on that, if there's any correlation there.
Yeah, so that's a very interesting question. I'm not aware of the data that you cited, but there is a very intriguing connection between PTSD for returning soldiers and the phenomena that I was talking about. And to make that connection, let me switch gears slightly. Instead of talking about nursing and foraging, let me talk about colony defense. So bees defend their hive from intruders. One of the main ways they do that is by stinging. So we're all familiar with bee stings. Bees, honeybees, deploy their stings defensively to defend their hive from attack. A honeybee colony is a very attractive target for attack because they have large quantities of really good food, honey, pollen, and for many organisms, the baby bees themselves are very attractive. Brood, for example, honeybee brood is a delicacy in Asia, as prized as a food. And it's certainly prized even more by ants, who will attack beehives and then other larger animals. So bees have evolved a very sophisticated defense system. They have guards at the entrance that alert the other bees inside when they detect an intruder. And the way they communicate to alert the other bees is another one of these chemicals or pheromones that says, hey, there's something at the entrance that doesn't belong here, whether it's a foreign honeybee from another colony or ants or a bear. It starts with that alert. And then there are a group of specialized bees called soldiers who go out once they are alerted and look for the intruder. And they're the first ones to attempt to sting the intruder, to repel it so that it does not further attack the colony. So we've studied gene activity changes associated with colony defense, exactly in the ways that I just described before for nurses and foragers. But we found something that's really relevant to your very interesting question. So we do assays in the laboratory that simulate a colony attack. We have dishes of bees, 10 bees in a dish. They're all from the same hive. And we put an intruder into that dish, a bee from another colony. Bees, even in the laboratory, even just with 10 bees, they can tell if the bee that was put in is from their own hive, meaning it's related to them, part of the family, or a foreign bee. And if it's a foreign bee, they will attack that individual the way that would happen at the entrance to a real hive. So we do those experiments, and the bees immediately attack the intruder. And then we note which bees have attacked the intruder. And then we wait about 20, 30 minutes, collect them, and then measure their gene activity. So I'll repeat what we did. We put the foreign bee in, the bees immediately respond and sting the intruder. And then we get the bees that responded most strongly, and we look at their gene expression. And we see that they show big differences in gene activity compared to when we have bees that have not been disturbed in this way. So one might ask, well, the gene activity changes that occurred 30 minutes later, by definition, can't have anything to do with the decision of whether to attack that foreign bee or not, because that happened already. And gene expression actually is slow. It's slower than changes in neural activity. That's why we wait 30 minutes. Changes in gene activity don't occur in milliseconds. They have to unfold over minutes. We wait 30 minutes to get a nice, clear, robust response, but it takes at least minutes to see a change in gene expression. Meanwhile, you can't wait minutes to respond to an existential threat. You have to respond immediately. So what are those changes in gene activity 30 minutes later doing? Well, we pondered that, and we developed a concept, and then we've tested that empirically. And the concept is basically like this. Neurons are for today, genes are for tomorrow. So the idea is there are two control systems in the brain, the neural control system and the molecular control system. The neural control system refers to the tuning of the neurons, sensory neurons, processing neurons, how sensitive they are, how quick they respond. And that's what happens when you have a disturbance. The neural system kicks right in, and it has to, because survival depends on an immediate response. So the neural system kicks in, the bee responds, detects the intruder, and does the aggressive behavior. Now what? Well, ecology predicts, and we're getting closer now to your connection, ecology would predict that a threat today means there might be a threat a few hours later, or even a threat tomorrow. That's how it works. If you're under threat, as an organism, you might continue to be under threat. So the response to an intruder actually triggers two things. The presence of an intruder actually triggers two responses. One is the immediate one, with the neurons firing and orchestrating the behavior. But all of that neural activity also triggers changes in gene expression to put the bee in a more alert state. In a more alert state, they are then going to respond more quickly to a disturbance. So I don't know if you or any of your colleagues in the group are beekeepers or know any beekeepers. If you do, basically all I've said is if you've disturbed a colony, you should stay away from it for a few hours because the bees are still aroused. They need to calm down. That period of time when they are alert needs to pass. How long is that period? Well, it depends on individual differences, on heredity, on experience, on the current environment, and so forth. So sometimes it might be a couple hours, and then the bees will calm down. Sometimes it might be a whole day. Well, PTSD, the threat never goes away. The threat in the brain never goes away. The individual is on alert. So there are epigenetic changes, we know, switching now from bees to humans, that keep a heightened alert state in the brain. It naturally goes away in bees, in humans as well. If you're startled, disturbed, you might be riled up for a little bit, but it goes away. PTSD, it does not go away. So there's a definite connection between environmental modulation of brain gene expression and what the most extreme manifestations of that might be.
Absolutely fascinating. And in the paper that I alluded to during the introduction, you recently authored a study that pioneers a non-invasive MRI-based imaging approach, which directly captures methylation within the brain. Dr. Robinson, could you please explain how this works exactly?
Sure. So first, I'll just say that the motivation for this is when we made these discoveries in honeybees about how sensitive the genome is to environmental information in the brain, and then others have made those discoveries in other animal species. Of course, there was a strong interest in extending the work to humans. But unfortunately, direct measurements of gene activity are not possible in live humans because the method of doing so is very invasive. So it's not possible to do it with humans. So myself and several colleagues, and by the way, this is a great collaboration that I am privileged to be part of, so not a solo effort at all. We started talking about how might there be ways of getting some information on how the environment affects the human genome in the brain, doing so in real time, live humans in a non-invasive way. So when you think about imaging with humans non-invasively, you think about MRI, magnetic resonance imaging, which uses a variety of different kinds of signals and then detectors to pick up the kinds of signals and then locate them in space in particular brain regions. So you can talk about activated regions of the brain based on how the signaling is interpreted. So we started thinking about those kinds of approaches and came upon a method that allows us to provide a completely harmless substance to people that would label a key molecule associated with epigenetics, which is a chemical called a methyl group that plays a key role in methylation, which is the addition of a methyl group to the DNA, and which is known to affect the expression of a gene. When the methyl group is present or when the methyl group is absent, it changes the expression of the gene. And we found that there's a method that's completely harmless, non-invasive, that can be used to label the methyl groups. And then we set out to test whether a certain kind of MRI system could be able to be tuned and developed to detect that methylation specifically on DNA and not methylation anywhere else. And the method has been looking very promising. We're still in the stage where we're using animal models in order to test the method, make sure it's completely safe, make sure it's helpful, instructive, gives good, consistent information. And because imaging, the bigger the subject, the easier it is to image. This is one case where rather than using mice, which are the standard models for most studies that are done in the laboratory with animals that are of potential interest to translating to humans, we're actually working with a pig model because the pig brain is roughly the size of the human brain. So when we put a pig in an imaging machine and we get the results, we know that's more comparable to the human results. So we've shown that we can give the pig a cocktail that labels the methyl groups. We've showed that the methyl groups then get labeled in the brain, the DNA gets labeled by the methyl groups attaching to that, and then searching for those methylated regions, we can then look at brain region differences in methylation in pigs, and we have a few more experiments to do before we will move to humans.
Fascinating. And what are the implications of being able to observe these epigenetic changes using MRI technology?
The implications are we will get a window onto changes in epigenetics in humans in real time. Studies have been done using blood as markers instead of the brain, because you can get blood non-invasively. And studies have been done in autopsies direct from the brain. And in both cases, they indicate that changes in DNA methylation are associated with changes in humans, various kinds of disease states, as well as traumatic states. But both blood and autopsy, obviously very limited. Blood is not brain. We want a readout of the brain. And of course, autopsy, very limited. So we will get a window on how the environment gets under the skin. For example, drug addiction is known to have many epigenetic changes. We would hope to be able to visualize that in real time. Alzheimer's, other changes, other states, as well as socioeconomic and stress and other forms. Given how we know changes in the environment affect changes in gene expression, epigenetics is one way to stabilize big changes in gene expression, and so could then be the target.
Fascinating. Could you explain why some epigenetic changes are short-lived while others may last a lifetime?
Yeah, so the explanations can be at two levels. One, at a high level of just functioning of the organism and the adaptiveness of those changes. One can imagine that one wants a range. So exposure to one environmental stimulus, you might want that, quote, recorded for a period of time, but not forever, because the environment changes. Whereas other kinds of stimuli might trigger such a strong response that it gets embedded for a longer period of time. And then, of course, we get into pathologies and things of that nature that may have such a strong effect that it just happens, that it's a more or less permanent effect.
And what role do epigenetics play specifically within the brain? So given that adult neuron cells do not divide, how does this differ from epigenetic processes in other parts of the body?
Well, epigenetics gives a level of plasticity to the brain, which seems to be very essential. The brain is kind of where the rubber meets the road, in a sense, how the environmental signals are received, interpreted, and then used as the basis for learning and memory or for changes in behavior and so forth. So having this ability to modify DNA, which will then modify gene expression, is a very important part of the brain's plasticity. And when you couple that with the fact that, as you say, that the brain, most parts of the brain, there are not cells that divide. There actually are some parts that do have the birth of new neurons. But overall, I think with the brain, the more forms of plasticity, the better.
Fascinating. And finally, Dr. Robinson, as you mentioned earlier, the field of epigenomics and also epigenetic editing is really burgeoning these days. And there are a number of biotech companies such as Chroma Medicine and Tune Therapeutics, which I'm sure you're aware of. How do you foresee the technology that your team is pioneering could perhaps be used in respect to the different epigenetic editing platforms that the aforementioned companies are developing?
I think that companies and other researchers could find it a very useful tool to be able to measure the efficacy of the treatments that they are developing. So looking at that, we are not working at the individual gene level. just like MRI does not work on the individual neuron level. The level that we're working at is at the brain region level. So that's exactly what MRI does as well. So that means the sort of aggregated signal of many, many, many cells in the brain. That's the readout of what MRI provides. So you take some of these methods to change epigenetics, epigenetic markings in the brain. This could provide one readout of those at a non-invasive level and at a high sort of brain region level.
Fascinating. Well, thank you so much for your time, Dr. Robinson. That was a very interesting conversation. And thank you for the work that you're doing. I know that there are many people in our community and other communities that are very excited about what your team are pioneering. So thank you for your time. And it was extremely fascinating.
Great. It's a pleasure to be here and nice to see you again. Take care. Bye-bye.
Thank you, Dr. Robinson.